Welcome to everybody today. Another wonderful day ahead. And it's a great pleasure to welcome Martin Deschens to give plenary lecture on genetics of stroke and VCI. I think Martin will be known to many people. He's director of the Institute of Stroke and Dementia Research at LMU in Munich. And he's got a long-standing interest in genetics. I'm not sure what he's going to talk about today. He's got a lot of interest in polygenic genetics, but also some interesting recent work on HGRA1 and the sort of interaction between monogenic and polygenic contributions to disease, which is really exciting. So I'm very much looking forward to hearing Martin talk, and welcome, Martin.
Thank you, Hugh, and thank you very much to the organizers. Good morning, everybody. Thank you very much for the invitation. A great pleasure to be here. I would like to give a brief overview on where genetics has taken us, primarily focusing on stroke and cerebral small vessel disease, but also touching on cognition at the very end. So more than any other field, I think genetics is transforming medicine. Genetics has provided us with numerous risk genes, offering key insights into biology and disease mechanisms. It allows clarifying the causal role of risk factors or exposures, has improved prognostication, facilitated drug development, changed disease classification, and we are seeing more and more examples where genetics improves treatment options. Now, the potential for drug discovery is also illustrated by megastroke, the so far largest stroke GWAS involving individuals of multiple ancestries. And this Manhattan plot, I'm sure you're familiar with it, provides an overview of the 32 genome-wide significant loci, 22 of which were novel, and many of which could be contributed to individual etiological stroke subtypes. Now, these loci were significantly enriched for known drug targets for stroke therapy, and this is really why I'm showing this. Two of the genes are targets of approved antithrombotic drugs, including alteplase, tenecteplase, and other thrombolytic agents, and cilostazol, an antiplatelet drug approved for stroke prevention in Asia, raising confidence, and this is the key point here, that our GWAS signals might harbor additional therapeutic targets. This panel here illustrates the discovery chain for HDEC9, a major risk locus for atherosclerotic phenotypes, including also coronary artery disease and peripheral artery disease, that emerged from our previous GWASs and that enabled us to go from locus identification to expression quantitative traits, gene regulatory mechanisms, animal models, and eventually pharmacological interventions. And just to give you a glimpse of it, what we found is that histone deacetylase 9, an HDEC that mostly localizes to the cytoplasm, binds to IKK alpha and beta, both key molecules in NF-kappa B signaling, thus resulting in the deacetylation and subsequent activation, which in turn then drives inflammatory responses in macrophages and endothelial cells. We further found that pharmacological inhibition of class 2A HDECs with TMP195 attenuates all these steps, including lesion formation in atherogenic mice and cytokine production in monocytes from patients. 4 weeks of treatment, it's just a small piece of the data, with TMP195 resulted in a significant reduction of early lesion sizes. What we found is that a reduction in endothelial cell activation and a reduction in the invasion of classical monocytes and leukocytes, neutrophils, into atherosclerotic lesions, likely as a consequence of impaired leukocyte endothelial interactions, as is shown here by intravital microscopy of the carotid artery. Treatment with the HDEC inhibitor further had a stabilizing effect on atherosclerotic plaques, for instance, by reducing here plaque or enhancing plaque thickness. And we are currently trying to optimize the treatment approach and drug delivery. However, for the purpose of this talk, I would rather focus on cerebral small vessel disease as the main driver of vascular cognitive impairment. Probably the strongest and most direct proof of vascular dementia, of the vascular dementia concept, comes from CADASIL. As you know, a hereditary condition caused by mutations in Notch3. So let's turn to monogenic conditions. Aside from CADASIL, there is an expanding spectrum of hereditary conditions caused by highly penetrant mutations, including CARASIL, to which I will get in a minute, CARASAL, a condition related to cathepsin A mutations, COL4A1A2 related angiopathies, Fabry disease, and an expanding spectrum of additional conditions. Now, A converging theme that has emerged from these conditions is a dysregulated extracellular matrix, as is most obvious for mutations in collagen 4A1, A2, both structural constituents of the ECM, mutations in Notch3, which accumulates in the extracellular matrix and recruits other bioactive proteins such as vitronectin and TIM3, and mutations in HTRA1, a serine protease. As it turns out, and as Hugh mentioned, HTRA1 is becoming a key player in cerebral small vessel disease. HTRA1 encodes high temperature requirement protease 1. It was originally identified as the gene underlying carosil, an exceptionally rare condition that was initially identified or seen in consanguineous families from Japan. We haven't seen those families in Germany here so far. These patients mostly present with lacunar stroke or vascular dementia, along with typical imaging features of small vessel disease and also some systemic manifestations, such as alopecia. Most of them present at young age, so between 20 and 40 years. The spectrum of mutations is broad. Most of them affect the protease domain. Please keep this in mind. Now, as an interesting observation, it subsequently became clear that heterozygous mutations in the very same gene may likewise become heterozygous mutation carriers may likewise become symptomatic. These patients typically have a much milder phenotype and present at higher age, pointing towards haploinsufficiency as a likely mechanism. And in fact, in most instances, heterozygous mutation carriers are phenotypically indistinguishable from sporadic patients, which makes this condition, of course, particularly interesting. Just briefly on the molecular mechanism, we found that HTR is involved in TGF-beta signaling. We found that HTR1 is secreted into the extracellular space where it associates with the ECM, and showed that HTR1 cleaves LTBP1, latent TGF-beta binding protein 1, thus liberating TGF-beta, an important signaling molecule. Now, carosil mutations are loss of function mutations, and as a consequence, LTBP1 is retained in the ECM, not processed, and so TGF-beta signaling is attenuated. But as you will see in a minute, these mutations really have a much wider range of molecular consequences. It has further become clear that HTR1 is also implicated in the more common sporadic forms of small vessel disease, as evidenced by both common and rare variant association studies. These are whole exome sequencing data from more than 16,000 individuals from the UK Biobank. And what we looked for here was rare genetic variants with an influence on white matter hyperintensity volume. What we found is that HTR1 is the top signal in gene burden tests. Domain-specific burden tests showed that the association was restricted to the protease domain, to variants in the protease domain. What you see here are representative MRIs from mutation carriers in red and matched controls. The frequency of such variants in the UK Biobank, so in the general population, was 1 in 450 individuals, so actually quite high. And as expected, there's an increase in white matter hyperintensity volumes with age. On average, the presence of an HTR1 mutation accelerated the phenotype by about 11 years. Note that this here is all on the log scale. What you see here is the effect size of protease domain variant carrier status and white matter hyperintensity volume in comparison with established risk factors. Protease domain variant carrier status had a substantially larger effect than hypertension, diabetes, BMI, ever smoking. Also, effect size was larger than for individuals with a polygenic risk score based on common genetic variants above the top percentile of the whole PRS distribution. So again, effect sizes are really considerable. We further examined the functional consequences of HTR1 variants in the protease activity assays using human cell-derived recombinant proteins and LTBP1 as a physiological substrate. Down here, the dotted line marks the wild type. This is the active site mutant. These are the carosil variants, mutations in the casal and PDZ domain, so outside the protein domain, showed normal protease activity, whereas the majority of protease domain mutants showed marked reductions in protease activity. Given these observations, we next performed a targeted analysis of gene candidate substrates of HTR1. And these are proteomics data now from mouse, from an HTR1 knockout mouse that we had generated in the previous study, in which showed a clear enrichment of multiple proteins in the brain vasculature. So this is the proteome of the isolated brain vasculature. of these mice. Among them, several known HTRA1 substrates, including, for instance, vitronectin and TIM3, which also are relevant in the context of CADASIL. Among the most strongly and significantly enriched proteins, we noted EGFL8. And interestingly, EGFL8 reached genome-wide, experiment-wide significance for association with white matter hyperintensity volume in the UK Biobank, suggesting that this molecule might fall into a joint pathway with HTR1 and itself may be relevant for small vessel disease. And indeed, incubation of human EGFL8 with HTR1, so the protease, resulted in a dose-dependent degradation of EGFL8 that was prevented by the HTR1 inhibitor. So this molecule is a novel HTR1 substrate, predominantly localizes to smooth muscle cells in the vasculature, so is expressed within arteries and arterioles. Its biological function is currently unknown, but EGFL7, a close related paralog, is likewise expressed in the brain vasculature and has been implicated in multiple vascular functions, including elastogenesis. I think I skipped this just to mention that these variants are also associated with other phenotypes, including migraine, which is interesting. But in the interest of time, I will now move to this aspect, which is a potential role of HTI1 in CADASIL, another type of hereditary small vessel disease. As mentioned, we previously found that the microvascular proteome of CADASIL, this is, no, I didn't mention this, has an HTI1 loss of function signature. These are isolated brain vessels from CADASIL patients, so not from mouse, but from patients with CADASIL, showing accumulation of Notch3 in granular osmophilic deposits here in immunohistochemistry shown in red. And these are the proteomics data from these vessels, showing that aside from Notch3, which expectedly is the top signal in this plot, are several other proteins that accumulate in blood vessels from CADASIL patients, including HTRA1. Now, HTR1 co-localizes with the Notch3 deposit, suggesting that HTR1 is recruited to these Notch3 aggregates. So we then went back to our knockout mice, and this is again the microvascular proteome of the HTR1 knockout animals, which I've shown earlier. And intriguingly, many of the enriched proteins overlap with those found in CADASIL patients and are substrates of HTRA1, as exemplified here, for instance, for c-MYC. And we've expanded this approach to other proteins, including semaphorin 3G, vitronectin, and OFML3. All these molecules are substrates of HTR1 and accumulate in the vasculature of HTR1 knockout mice and CADASIL patients. So the proteomic profile of CADASIL blood vessels has an HTR1 loss of function signature. And our hypothesis really is that recruitment of HTR1 into the Notch3 aggregates results in inactivation or lack of availability of HTR1, causing a loss of function. Whether this contributes to disease pathogenesis in CADASIL patients, we currently don't know. What is clear, however, that HTR1 is a central player in small vessel disease in accordance with its biological function of regulating multiple constituents of the extracellular matrix, including LTBP1, vitronectin, TIM3, EGFL8, and so on. Still, the ECM isn't the only mechanism for which there is evidence from genetics, which brings me to my next slide. What you see here are rare and common variants associated with small vessel stroke. Some of these genes, including HTR1 and COL4A1A2, are implicated in both rare and common genetic variant associations, so sporadic disease. And several of these genes have established roles in other functions, including blood-brain barrier formation, as evidenced, for instance, by collagen 4A1 and FOXF2, another gene implicated in small vessel stroke. Genetics further points to an involvement of endothelial cells and pericytes, but also smooth muscle cells. So we have a whole bunch of mechanisms on several levels affecting multiple constituents of the neurovascular unit. So a lot to do for the next years. Now, so far I've been mostly speaking about monogenic small vessel disease. I will now focus on common genetic variants as a starting point for discovery. And this Manhattan plot is from a recent transethnic GWAS on lacunar stroke that was coordinated by Hugh Marcus and that further integrated GWAS data on white matter hyperintensities in a multi-trait analysis. The red asterisks up here mark loci that reach genome-wide significance in the more restricted analysis in lacunar stroke. So many of these signals here come from a combined phenotype with white matter hyperintensities. Just to note that HTI1 is again on the list. And this figure is from a more targeted GWAS on white matter hyperintensity volumes in DTI measures that was coordinated by Stephanie de Wet and found 27 loci influencing white matter hyperintensity volume. Slightly different way of displaying a GWAS here in a Manhattan plot. What you can see here is that there's substantial genetic overlap with other traits and risk factors, in particular blood pressure traits, so systolic, diastolic pressure and pulse pressure, but also BMI, type 2 diabetes, and also Alzheimer's disease. Several of the prioritized risk genes in these regions represent registered targets of approved drugs, again emphasizing the potential for drug discovery and preventive treatment. Now, the strongest risk factor for all these phenotypes, of course, is, and it doesn't come as a surprise, age. And so we, in collaboration with Matthew Traylor, recently set out to perform a GWAS on age-related white matter hyperintensity or microstructural integrity as a major determinant of cognitive decline, dementia, and gait disorders. These analyses were based on DTI data from UK Biobank, and overall we identified 18 loci. Just want to dive into one of them. Among the top loci was a locus encoding the human major compatibility complex, and of the imputed HLA alleles and complement component 4 structural haplotype variants in the human MHC. The strongest association really was with the C4BS variant, which accounts for about 12% of all C4 structural haplotypes. And of course, complement is a really interesting target here in this context. The protective influence of the C4BS haplotype was stronger in elderly individuals and in subjects without a history of smoking. As you might know, complement has an important role in tissue homeostasis, in part through its influence on microglia, which we discussed yesterday. C4 influences synaptic remodeling, and it leads to increased microglial engulfment of synapses. Complement and complement receptor 3 are involved in spine elimination, in neurodegeneration. There's some very elegant work, for instance, from Katarina Akasoglu's lab, through binding of fibrinogen. So there is a potential link with increasing BBB permeability that is seen during aging. But let's go back to traditional risk factors. Genetics further opens the possibility to study the causal relationships between key exposures and outcomes using a technique called Mendelian randomization. And looking at small vessel disease and related stroke, you will not be surprised to see that both that blood pressure traits, type 2 diabetes, and so on, are causally involved in lacunar stroke, whereas BMI, there was no such relationship. Now, you can go one step further. Drug MR goes this one step further by restricting the analysis to genes that encode known drug targets, thus enabling the prediction of therapeutic interventions, specifically targeting a specific receptor, an ion channel, or other molecule. Here, for instance, we examined the effects of 3 different classes of antihypertensive agents: ACE inhibitors, beta blockers, calcium channel blockers, scaled to the respective effects in randomized controlled trials. This is coronary artery disease, this is stroke. And most notably, we found that blood pressure lowering through genetic variants in drug targets for calcium channel blockers and ACE inhibitors lowered stroke risk, whereas this was not the case for beta blockers. This is very reassuring, but probably not so novel and unexpected. But one of the attractive aspects of MR is that this approach can be expanded to settings where there are no available data from clinical trials, as is the case for etiological subtypes and small vessel disease. So here we analyzed MR associations between genetic proxies for antihypertensive drug classes and risk of stroke subtypes. And to make a long story short, what we found is that, again, calcium channel blockers are more effective than beta blockers, and this particularly applies for small vessel stroke and also matches with the results on white matter hyperintensities. So this, I think, could guide our treatment decisions in the absence of clinical trials data for small vessel stroke, and could be seen as another argument for choosing calcium channel blockers rather than beta blockers in patients with manifestations of small vessel disease. We further looked at the effects of blood lipids and lipid modifying treatments on phenotypes related to cerebral small vessel disease, in particular small vessel stroke and white matter hyperintensities. what we found is that genetically elevated HDL-C levels were associated with both lower risk of small vessel stroke and white matter hyperintensity stroke, and that this effect is primarily mediated through medium-sized HDL cholesterol. And most importantly, however, we found that elevated HDL-C levels through CETP inhibition is predicted to both lower risk of small vessel stroke and white matter hyperintensities. I should mention, though, that the beneficial effect of genetically determined HDL-C levels on small vessel stroke and white matter hyperintensities come at a price and are accompanied by an increased risk of intracerebral hemorrhage. So obviously, the net benefit of such an approach would need to be tested in a clinical trial. I'm coming to an end now. I've mostly spoken about stroke and small vessel disease, but what about vascular risk factors in midlife and incident dementia risk? How can genetics help us here? You know that the American Heart Association is propagating the concept of life's simple 7 as a set of risk factors that are accessible to interventions either through lifestyle modifications or pharmacological interventions. Here we use data from the UK Biobank, combining a longitudinal approach with a Mendelian randomization approach to look at the causal effects of individual biological and lifestyle risk factors. What these panels show here is the distribution of the overall score, the biological score and the lifestyle score in those with and without incident dementia. The observational data suggests that adherence to the biological metrics, that is blood pressure, cholesterol level, glycemic status, was associated with a lower risk of incident dementia, whereas there was no such relationship with lifestyle factors. In the Mendelian randomization analysis, genetically elevated blood pressure was associated with a higher risk of dementia. So all in all, these findings emphasize the importance of blood pressure control in midlife to mitigate dementia risk. Just as an outlook, genetics can also be leveraged, and I think you mentioned on this, to identify high-risk individuals. And in principle, this could be done from birth, allowing personalized approaches to screening and early intervention. Just to briefly highlight this aspect, this slide shows the hazard ratios for ischemic stroke, so not cognitive impairment, just ischemic stroke, depending on their genomic score, compared to individuals in the middle decile. Individuals in the top 0.25 percentile of the population are at a 3-fold increased risk of developing an ischemic stroke. Critical question, however, is, can this risk be modified? And indeed, it can be modified, as the data suggest. Here we integrated information on the GRS with information on risk factor levels based on recent treatment guidelines. And just looking at individuals in the top percentile of the meta-GRS who have either high levels of risk factors, here in red, the cumulative incidence of stroke by age 75 reached 8.5% in males and 5.1% in females. An effective reduction in the levels of the modifiable risk factors is predicted to result in a substantial risk reduction, so compensating for the high genomic risk. And this is my last slide. Conversely, for individuals matching the guidelines for established risk factors, we found notable differences for individuals in the top compared to the bottom percentile of the meta-GRS, further indicating that the meta-GRS captures residual risk not reflected by existing risk factors. So in principle, GRS for stroke could be used to identify individuals that might benefit from more intensive monitoring and early lifestyle interventions. To summarize, I hope I've been able to show that genetics not only provides us with numerous risk genes, but that these findings are offering insights into biology and mechanisms, help clarifying the causal role of risk factors or exposures, improve prognostication, facilitate drug development, and also eventually guide treatment decisions. So let me close not without thanking all the people involved, in particular Rainer Malik, Marius Georgakis, and other people from my lab, the Megastroke Consortium, in particular Stephanie DeBet, Hugh Marcus, Stephen Kittner, and Matthew Traylor, but also Kathy Sudlow, and our colleagues who did the proteomics here in our building. Thank you very much for your attention.
Thank you, Martin. That was a really superb overview, and it shows how exciting the data is that's coming out of genetics, and what the potential is. And it's an area we just need so many more people to work in to follow up some of these associations. I don't know whether we've got any questions. For some reason, my bar at the bottom is obscuring the questions. Let me see if I can. Are there any? Can you see any, Martin, on the chat?
I have to open the chat. Let me see.
Sorry, but for some, I can't get rid of my bar at the bottom. My
chat is empty.
Bar. But if not, if there aren't any, I've got some that I would like to ask. But can you?
There's nothing in the chat.
Okay, so I mean, so if people have got questions, just put up your hand or ask, and Raj will check on the bar, which I can't see, unfortunately. I mean, there's so many questions I could ask about it, but there are just a couple. I mean, you showed the very interesting data on HDL. What's your view on treating? I mean, you know, the data suggests that LDL doesn't really contribute to this disease. I mean, what's your, you know, do you think that should guide our treatment on statin therapy, or should we be doing trials in this area? It's always intrigued me, that sort of lack of association.
Yeah, obviously, that's a very important question. And I think, I mean, I don't think that the lipids data, the MR results by their own, should guide our treatment decisions in patients with small vessel disease. Definitely, we need clinical trials to explore that questions. I mean, I also need to emphasize that, of course, MR, like any other method, has its limitations. And the results definitely need to be interpreted in the context also of observational data and eventually clinical trials data, but also experimental data, which is what we call triangulation of evidence. So ideally, you would have both observational data, genetic data, and experimental data, all pointing in the very same direction as we do have, for instance, and that's data I didn't show, for MCP1, an inflammatory cytokine, and its causal involvement in large artery stroke. Regarding your question of lipid treatment, I don't think that there's any rationale at the moment to give CETP inhibitors to patients with small vessel disease on the basis of a single Mendelian randomization study. For one, CETP inhibitors haven't been shown to be beneficial in the larger population of patients with cardiovascular disease. Still, there's quite some ongoing discussion whether they should be given to these patients at all. So a decision would be much more difficult than for LDL lowering strategies. Second, we still haven't got a full oversight on balancing the risks with intracranial hemorrhage. As you've seen, the treatment and raising treatments would be expected to also raise the rate of intracranial hemorrhage. So we need to balance the effects. I think it's just too early. We could, and I wouldn't even be sure whether the pharmaceutical industry, on the basis of previous trials data on coronary artery disease, would be willing to invest in a trial in small vessel disease if there is a lack of signal in coronary artery disease.
And just one brief question, because I'm running out of time, but something I did want to ask you is, I mean, I think the HTRA1 data is really exciting, and, you know, the fact that it's extending into common sporadic stroke, you know, as well, really, you know, those sort of odds ratios you've got associated with variants in it were really large. Are there any sort of therapeutic strategies that, you know, might be available to target that pathway, or is that too far off at the moment?
I think it's a bit far off. We're currently working on HTR1. There's much more to tell regarding mechanisms. For instance, it's a trimer. So there are three identical molecules that have to come together in order to gain activation as a protease. And some of these mutations, data I haven't shown, interfere with the assembly, with the trimerization. We have developed approaches to reconstitute trimerization, and by that reactivate the protease. This is all work in mice, but in humans, it will be difficult, at least in my view, at the moment, without more advanced, for instance, genetics and viral approaches to target HTR1, because first of all, HTR1 is expressed by multiple cell types in the brain, including and in particular astrocytes. So you would need to get it into the brain, first challenge, but also you want to avoid off-target effects, which is always an issue. Just giving HTR1, injecting it, wouldn't do the job. So I think we still need to work on this, particularly using animal models. But yes, I mean, yeah.
There is a question from Chris Chen in the chat. Are there any interactions between small vessel and large vessel disease genes?
So yeah, Martin.
Yeah, I mean, always the question what you call an interaction. There is some overlap, of course, in the sense that there are genes. The most obvious example is genes implicated in hypertension. So in joint risk factors for large artery stroke and small vessel stroke. So there is a clear overlap or interaction, however you want to call it. Of course, then there is the complication that you may have additive effects on a symptomatic level, meaning that patients with a large artery stroke and in small vessel disease in addition might be more likely to come to attention and even be recruited into these types of studies. I'm not aware of any particular genes where there would be a molecular interaction between a gene implicated in large artery stroke on the one side and another gene being implicated in small vessel stroke. Also, just to mention that we don't have so many genes for small vessel lacunar stroke so far.
Okay, thank you, Martin. I think we better stop there because we need to keep the time, but thanks again, a really wonderful talk and lots of exciting things to think about and plan in the future. So I'm now going to pass over to the first session of the day. So back to you, Raj.